This training is required for anyone working with recombinant or synthetic nucleic acid molecules to comply with the National Institutes of Health guidelines for research involving recombinant or synthetic nucleic acid molecules, the Center for Disease Control and Prevention, and other regulatory agencies. The NIH guidelines describe the practices for working with recombinant and synthetic nucleic acid molecules. Examples include recombinant DNA, polymerase chain reaction, and synthetic viruses. Research involving recombinant and synthetic nucleic acid molecules must be approved by the university's Institutional Biosafety Committee and follow the NIH guidelines for all funding resources. The university must have a biological safety officer in the Institutional Biosafety Committee who is responsible for managing risks associated with recombinant or synthetic nucleic acid molecules. They also must conduct inspections of labs and report issues and incidents. The principal investigator is responsible for full compliance of the NIH guidelines. A principal investigator has 7 areas of responsibility. 1. Determine the sections of the NIH guidelines applicable to the research. 2. Obtain IBC approval before beginning work. 3. Assure all research staff is current on required training. 4. Adhere to IBC-approved containment requirements. 5. Adhere to IBC-approved safety procedures and emergency plans. 6. Comply with shipping requirements for recombinant or synthetic nucleic acid molecules. 7. Report any incidents to EHS immediately. There are 6 categories of experiments outlined in Section 3 of the NIH guidelines. Section 3A covers experiments that require NIH director and IBC approval before initiation and are considered major actions. For example, the deliberate transfer of a drug resistance trait to microorganisms that are not known to acquire the trait naturally, if such acquisition could compromise the use of the drug to control disease agents in humans, veterinary medicine, or agriculture. Section 3B covers experiments that require NIH Office of Science Policy and IBC approval before initiation. Section 3B1 covers the deliberate formation of recombinant or synthetic nucleic acid molecules containing genes for the biosynthesis of highly toxic molecules lethal to vertebrates. Section 3B2 covers experiments that have been approved under Section 3A1A as major actions under the NIH guidelines. Section 3C covers experiments that require IBC approval and approval from all applicable institutional and regulatory authorities before initiation. Human gene transfer, deliberate transfer into human research participants of either recombinant nucleic acid molecules or DNA or RNA derived from recombinant nucleic acid molecules or synthetic nucleic acid molecules or DNA or RNA derived from synthetic nucleic acid molecules that meet any one of the following criteria: contain more than 100 nucleotides, or possesses biological properties that enable integration into the genome, for example, cis elements involved in integration, or have the potential to replicate in a cell, or can be translated or transcribed. Section 3D covers experiments that require Institutional Biosafety Committee approval before initiation. Most university research is Section 3D classified under one of 7 subcategories. Principal investigators must be familiar with their research category for compliance. Section 3D1 covers experiments using Risk Group 2, Risk Group 3, Risk Group 4, or restricted agents as host vector systems, for example, using adenovirus, adenovirus luciferase, or adeno-associated virus to transfect cells. Section 3D2 covers experiments in which DNA from Risk Group 2, Risk Group 3, Risk Group 4, or restricted agents is cloned into non-pathogenic prokaryotic or lower eukaryotic host vector systems. For example, cloning Brucella genes into K12 lineage non-pathogenic E. coli. Section 3D3 covers experiments involving the use of infectious DNA or RNA viruses or defective DNA or RNA viruses in the presence of helper virus in tissue culture systems. For example, the creation of recombinant vectors. Section 3D4 covers experiments involving whole animals in which the animal's genome has been altered by stable introduction of recombinant or synthetic nucleic acids into germline, transgenic animals, or viable recombinant or synthetic nucleic acid molecule-modified microorganisms are tested on whole animals. Section 3D5 includes experiments involving whole plants in which plants are genetically engineered by recombinant or synthetic nucleic acid molecule methods, or plants are used with recombinant or synthetic nucleic acid molecule containing insects. For example, plants associated with exotic infectious agents or associated with serious environmental concerns. Section 3D6 covers large-scale experiments involving more than 10 liters of culture containing recombinant or synthetic nucleic acid molecules. Section 3D7 covers experiments involving influenza viruses. Experiments with influenza viruses generated by recombinant or synthetic methods shall be conducted at the biosafety level of containment corresponding to the risk group of the virus that was the source of the majority of the segments in the recombinant or synthetic virus. Experiments with influenza viruses containing genes or segments from 1918 to 1919, H1N1, 1918, H1N1, human H2N2, 1957 to 1968, and highly pathogenic avian influenza H5N1 strains within the goose Guangdong 96-like H5 lineage, HPAI H5N1, including but not limited to strains of HPAI H5N1 virus that are transmissible among mammals by respiratory droplets, as demonstrated in the appropriate animal model or clinically in humans, hereinafter referred to as mammalian transmissible HPAI H5N1 virus, shall be conducted at biosafety level 3 enhanced containment. If your experiment is not covered by any of the sections mentioned but is covered in Section 3E, IBC approval is required. The university's requirement exceeds the NIH guidelines. You must follow all university requirements. Section 3E covers experiments that require IBC notice simultaneous with initiation. Section 3E1 covers experiments involving the formation of recombinant or synthetic nucleic acid molecules containing no more than 2/3 of the genome of any eukaryotic virus. Section 3E2 covers experiments involving nucleic acid molecule modified whole plants and experiments involving recombinant or synthetic nucleic acid molecule modified organisms associated with whole plants, except those that fall under Section 3A, 3B, 3D, or 3F. For example, corn-expressing genes from the Bacillus thuringiensis or tobacco plants expressing green fluorescent protein. Section 3E3 covers experiments involving the generation of transgenic rodents, such as experiments that require BSL-1 containment. Experiments that require BSL-2, BSL-3, or BSL-4 containment are covered under Section 3D4. Section 3F is classified as an exempt experiment under the NIH guidelines. The university requires approval by the Institutional Biosafety Committee for all exempt experiments. To receive credit for this training, complete the test with a score of at least 80%. For questions or assistance, contact EHS.